Hello and welcome to this Nature.com custom webcast titled Organoids as a Model for Studying Pathogen Infection, including SARS-CoV-2. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Nikon. We'll begin the webcast with two presentations. The first is from Dr. Steve Boulon, Group Leader in the Department of Infectious Diseases Virology at Heidelberg University.
And that can pneumonia. But there is actually increasing evidence that there is a systemic manifestation of SARS-CoV-2 brain and central nervous system symptoms by the presence of stroke, seizure. Many patients are reporting gastrointestinal issues like diarrhea, has been kidney damage reported, liver damages, blood a bit through the body, and also heart inflammation and damage to the heart muscles. So we just actually want to understand SARS-CoV-2 life cycle, not only in the lung, but also organs. And why do we actually care? That's actually appear very obvious, but the lung is going to be from the gut. If we actually look at the immune system in the lung or in the gut, these are going to be very different, and they will respond differently. The difference in viral replication and de novo virus is different between the two organs. And importantly, the mechanisms of pathogenesis are going to be very long and when we look at a single organ, we also have to consider the different cell types that there is in each individual organ. If the lung on the right side, you can see that there is different cells in the top or the lower airway. And of the virus receptor, SAE2, or the cellular protease compress 2, different in the different cell type. And there's actually some work now has been done extensively to type express the receptor and the cellular protease compress 2 by many groups now throughout the world. But what is actually very important here is like the A549 cell, one of the main cell line which is used as a human lung model, depending they have no SAE2, little amount of SAE. So it really good to insist of the importance of using primary cells versus transformed cells. I think it's common knowledge to know that transformed cells have a defective immune pathway, but remember that. But also, transformed cells are going to react differently to drugs. A good example for that is anticoagulants act very differently between the cell line and primary tumor tissues. And more recently, with all the work which is currently being done on SARS-CoV-2, we also realized that antiviral compounds act very different to whether you are looking at 549 and KLU3 cells, both lung cells, but some protects against SARS-2 in A549 cells, but not in KLU cells. Consider that in the model system. And a good example of this is some work we have performed in my lab where we have compared human colon carcinoma derived cells, like colon organoids derived from patient isolated stem cells, where organoid with interferon measure the antiviral response by actually of interferon stimulated G. You have the example of IFIT1, and you can see colon organoids are much more responsive compared to the T84 colon immortalized line. So it's the importance of using primary. About using primary cells is now, sorry, I've clicked one more, is that we organoids which are present for all most organs which are actually targeted or impacting the function impacted by SARS-CoV kidney organoid, that you can also hear this by Nuria Montserrat in the other lecture. And her organoids are also available to study SARS-CoV-2 infection replication. Here in group is what happened in the gut. So what is the importance of the enteric phase of SARS-CoV-2? So why do we care about the gastrointestinal tract? Actually, from the beginning of pandemic, we found that patients have gastrointestinal. So a fraction between 20 and 30% have diarrhea episode. There is also a fraction of patients that have genome, SARS-CoV-2 genome detected in their feces, but we yet don't know whether they are actually infectious virus particles in the stool right now. Recently, some patients keep shedding SARS-CoV-2 genome in their feces while being tested negative by oropharyngeal swab. That could actually suggest that the gastrointestinal tract is actually representing a reserve of virus, a reservoir, for the virus in the patient. And evidence that SARS-CoV-2 replicates in the GI tract because there has been biopsy work that clearly show replicating virus in the human intestinal epithelium. As I said, there have been a few reports that there is infectious virus particle in the feces, but there is very limited report with this, and address that in a much more systematic manner. Look at the intestinal epithelium. It's a complex phase, composed of multiple cell types, organized as a monolayer of cells, literally separating us from the outside environment. These cells are in constant contact with the ever-present commensal flora, and they tolerate the presence of the flora while remaining responsive against enteric pathogen. This regulation of these fine difference between tolerance and immune response can lead to inflammation, inflammatory bowel disease, and even cancer development. So, if we want to look at how gut homeostasis is a complex interplay between the intestinal epithelial cells and a lot of very specific immune in the lamina propria, which regulate production of mucus, production of antimicrobial peptides, but also cell proliferation and cell differentiation. But here today I'm going to on the very first response that every cell do during pathogen challenge, which is called intrinsic innate immune response. And in the virus, when the virus enters the cell, it is sensed by a pathogen recognition receptor, which will induce the production of cytokine and interferon. And in the case of the gut, there is two important cytokines which are made, the type 1 and the type 3 interferon. Autocrine and the parine manner produce or to induce the production of interferon stimuli gene, which will create an antiviral state into the cells. So in February, over the years, my lab and many other labs have actually worked out that the type 3 interferon, which is very specific to epithelial phases, is the first response against enteric virus in the gut. While if infection persists and start to be systemic, immune cell will be involved and the type 1 interferon will act as an antiviral. So when the infection started to come in Germany, so basically at the end of February, we started to get our first patient, isolated the virus from nasopharyngeal and oropharyngeal swab, and grew them like everything else in a viral cell system. And the first thing we decided to do is to look whether colon carcinoma cell line, so the KCO2 cells, can actually support SARS-CoV-2 replication and production of de novo virus particles. So what you see here is a time course of infection where cells are stained at different time post-infection using an antibody directed against the nucleocapsid N of SARS-CoV-2 and against a double-stranded RNA. It's using an antibody called J2, and double-stranded RNA are an intermediate of replication. So when you see a signal, it's very specific for any virus, RNA virus replication. So we could see that within 4 hours, we can detect actually production of de novo protein, viral protein, and within 6 to 8 hours, we even have de novo virus production. So it's a very fast set of infection. So we then went to an organoid model where, as opposed to Nuria, where she used pluripotent stem cells, we basically isolate tissue-specific stem cells from the gastrointestinal tract, and we take this stem cell containing crypt, grow them in differentiation medium, and to actually grow our organoid model. And again, the advantage of the organoid is that they are primary, non-transformed human intestinal epithelium cells. In an intestinal organoid, have all cell types that are naturally present in the human gut, or most cell type, I should say. These cells are genetically modifiable, and they are patient-specific, so meaning we actually even can do personalized medicine approach. So the first things we look at our organoids, so validated that they are differentiated, so we could stain them for cadherin at adherence junction. So you can here see an organoid and in fluorescent staining kind of through one of the villi lecture. We can identify mucin-positive cells marking goblet and also enteroendocrine cells. We also use RT-PCR for differentiation when we started the differentiation procedure, and we can here look at a stem cell marker, at a marker for enterocytes, so that's sucrose isomaltase, or mucin as a marker of goblet cell, and you can see that during differentiation, we are really increasing the amount of the different cell type that we naturally have into our organoid model. We now look at this organoid and tested whether they express SAE2 and the cellular protease TMPRSS2. And you can actually here see that human colon-derived organoid express much more SAE2 than other cells that we have tested, even the long cell LU3 cells. And they also express a relatively large amount of TMPRSS2 that we can see both by qRT-PCR and by Western blot analysis. So we are technically having all the machinery to infect this organoid with SARS-CoV-2. We could now perform the infection, and we decided to work not in 3 dimensions like we normally do with organoid, but in 2 dimensions to basically improve the pipeline and also perform a more high throughput approach to it. We grew within 2D and them with SARS-CoV-2, and again, you can clearly detect infected cells, so SARS-CoV-2 infected cells marked by the nucleocapsid N, by the presence of the double-stranded RNA, which are very specific for virus-infected cells using the J2 antibody. And we can really see that independent of the donor, so we use organoid coming from different patients, we do see about 10% of the cells being infected organoid. We can clearly see nice replication of increase of genome copy number. And most importantly, upon infection, we notice that these cells mount an immune response. And this immune response is characterized mostly by the production of type 3 interferon, which is also here known as interferon lambda. We saw a little induction of interferon beta, but it was mostly non-significant. This actually really shows that type 3 organoids are susceptible to SARS-CoV-2 infection, and they mount an interferon-mediated response upon infection. And this actually results that the results we obtain are actually completely in agreement with some work that was done by the lab of Hans Clevers in the same time as we were doing our work, where they performed RNA-seq on SARS-CoV-2 infected organoid. And I guess you probably cannot read here the name of the gene, but the signature response of interferon and interferon stimulated gene. And you can see here also production of interferon lambda in SARS-CoV-2 infected organoids. So the question we really wanted to address is that whether this interferon is able to protect organoid against infection. And for this, we set up experiments a little bit different, where we pre-treated our cell with interferon and then infected with the virus. And as you could expect, if we treat the organoid interferon, we are basically protecting the organoid against SARS-CoV-2 infection. So we are decreasing the number of infected cells, decreasing virus replication, and also significantly decreasing from several log decrease production of de novo virus particles. So the interferon response is actually very efficient in protecting human intestinal epithelium cells against SARS-CoV-2 infection. So basically what we have done during our first work, so the work we have done basically during the spring now, is we could show that intestinal organoids support SARS-CoV-2 replication and production of de novo virus particles. This organoid mount an immune response, which is characterized by production of type interferon, and this interferon partially protects the immune against SARS-CoV-2. Most importantly, we could see that de novo virus particles are released from the apical side of intestinal epithelium cells, which could provide an explanation of the presence of SARS-CoV-2 gene hIECs. And this is actually also in agreement with the work from Singh that actually have done the same action. Now I would like to finish the presentation by actually what we should now be doing to bring this organoid model to the next level to study host-pathogen interaction. What we are currently doing, because we have now many cell types in our organoid, is to use the organoids, infect them with the virus, and now look at the single cell level, what is the immune response generated by each individual type. So we do this using the platform, and we are seeing what is the immune response generated by each type and infected cells versus bystander cells. So this is currently work which is undergoing in my lab. We are finalizing the analysis, but what I can already tell you is we are comparing organoids which are derived from the ileum or the colon in patient. So you can here see the cell clustering of the different cell type that we have in our organ, and we can now where and which cell type is actually expressing ACE2. And you can here appreciate that there is ACE2 expressed at different level depending on the now, basically matching this to which cell is actually currently infected by SARS. As I said, this work is undergoing, so I'm not going to speculate too much on the results we are. These transcriptomic data are always very exciting, but I think they have one main problem. They do not allow us to actually see where it happened in space and also in time, because we just have a snapshot of the transcriptional profile of the different cell type. And that's why we decided to now actually use a single molecule RNA feed multiplex at very high level using the RNA-scope technology to now have where those infections takes place in an organoid and how it's happening at a function of time. And one of the main advantages of these techniques compared to classical immunostaining is that you are now looking at the transcript. You are not looking at a direct advantage. You want to validate transcriptomic data, because in transcriptomic data, what you get is transcript or RNA data, not necessarily a match between RNA and protein level, but most importantly, as all of you will know, often we do not have antibody which are specific against the protein we want to look at. So with this technique, we are actually able to bypass this limitation. So the way we do it is we infect the organoid and then slice them by cryosection, and then look at them by multiple rounds of RNA fish hybridization using 3 probes, with proliferating cells, stem cells, enterocyte markers, the ACE2 and the virus. As a proof of concept, I can show you the results we are having now is we are having able to look in the same organoid with different genes and basically correlate response which cell is actually infected by SARS-CoV-2 and which cell express TOMPRESS2 or A2. This can be within the same staining. And finally, what we should now be doing is consider the concept of time. How does this process happen in time? And for this, we should now start to focus on building organoid culture model that express various reporters that will tell us which cell is infected by SARS-CoV-2, which cells mount an immune response, and which cell is actually responding in as a bystander cells to, let's say, the cytokine. And the way we are doing it is we are using light sheet microscopy, and what you can see here is live light sheet microscopy movie. So you are going to fly within the 3 dimension of the organoid, and you can basically appreciate the complexity of the organoid, and I would say the beauty of the organoid. So by using this, we can now look at which cell is infected initially and how the infection is actually spreading in space, and correlate that response is spreading. And that's a setup that we are pushing quite a lot in the lab. So to conclude, so organoids, and I think we convince you that they are the perfect model to study SARS-CoV-2 infection or any pathogen, because they are primary model system with multiple cell type. They have they allow for the measurement of and correlation between various presence of or absence of replication or restriction factors by using single cell sequencing, for an example, and allow for they are very immunoresponsive, and they are also extremely good to study tissue-specific pathogenesis, but also to study pharmacokinetics of drug in a primary system that closely mimics organ-specific tissue. But the organoid, and I think this is important to highlight them, is actually their main advantage. They are multicellular and 3-dimensional objects, so that makes things much more complicated to infect or to look at. Their infections vary a lot with the differentiation protocol and how you differentiate. So you have to be extremely careful about this, and you have to consider where those infections take place. And this is very important for the lung gut, where the infection, the virus, come from the apical or the basolateral side. Finally, one thing that we really need to improve in an organoid, and I know many labs are working on, is that there is no immune cells. And this is something we need to integrate if we understand host-pathogen interaction as happening in the tissue. And in the case of the gut, in the lung, also the absence of microbiota, and we all know that cell behavior will be microbiota present. So with this, I would like to acknowledge my team and collaborator. Particularly, I would like to acknowledge Megan Stanifer from my group, which basically I've worked day and night pretty much every day for the last 3 months to generate this work, to actually do this work. And she's now supported by Camila, Carmon, and Patricio in my lab. I also would like to thank our collaborators, Theodore Alexandrov and Sergio Triana, with whom we are doing the single sequencing and multiplex RNA fish experiments. And finally, many other collaborators, but I would like to thank Ralph Schlager and Mirko Corte, because we actually started the work on SARS-CoV-2 all together in the department. With this, thank you for your attention.